going. So I'm going to put this virtual lab link into the classroom to go along with it. I figured that I'm going to make a video just in case access the lab. There's Flash, so there are certain devices that don't necessarily they allow Flash to work. So I'm going to do it here too. So it's up to you to watch this video, or if you would, totally up to you. But like be some questions involved. But yeah, with that, we'll just get started here. All right, so we're going to be isolating some DNA and then extracting it, and yeah, separating it basically. So that's going to be the purpose of the lab. So here are just some certain things that we can track with DNA. I'm sure some of you have seen some of these before. They do a lot of it on like CSI and stuff like that. But so here are just 3 reasons why it might be good to isolate DNA for scientists. So it could be for genetic testing, for identifying bodies, or for forensic evidence, like if there's a crime scene or something like that. They can extract DNA and get some clues from there. So DNA extraction is an important part of the process because it needs to be purified away from everything else that could be there. Yeah, just a fun fact. Inside each nucleus, there's about 2 meters of DNA. So if you think about how big a cell is already super tiny. The nucleus inside there is even smaller, and somehow there are 2 meters of DNA inside of that nucleus. It's crazy to think about. I don't know what it is about these chromosomes, but they sort of look like really abnormal looking fingers to me. They look like ogre fingers. So there you can see how it's spiraling out there. Come on. There we go. All right, so first we're going to take some cells from our person. There's our person. What we're going to be doing is we're going to be swabbing the inside of the cheek. So the skin on the inside of our mouths lose thousands of cells every day, and those are the cells we're going to be taking. So first thing we're going to do is we're going to collect the cells, then we're going to burst them open to get the DNA, then we're going to separate the DNA from everything else in there, and then we're going to isolate the DNA. So we have all of our materials here. There's a water bath, a centrifuge, some micropipettes, and a swab, and some other stuff. It's going really slow when you're trying to record this while running this at the same time. So I'm sorry. It would probably go faster if you do it on your own, if you are able to. Come on. Come on. Okay, so the first thing we're going to do is we're going to take the swab and swab his mouth. So our swab is covered with hundreds of tiny cheek cells, and inside each cheek cell is a nucleus, and inside that nucleus is where the DNA is. Then we're going to take some of the solution here, and we are going to put it into our thing, our little tubey thing here. So we're going to add some lysis solution, and lysis is a Greek word that means to separate. Then we're going to take our tube and we're going to put it into the bath here. There's the fingers again. And there's the unraveling there. So our solution that we added has two ingredients, detergent and proteinase K. So the detergent disrupts the cell membrane and nuclear envelope and causes the cells to burst open to release the DNA. And the DNA is still wrapped very tightly around proteins called, I don't know if it's histines or histones, and the proteinase K cuts apart the whatever that is to free the DNA. Alright, so next we're the DNA. So there's our little tubey thing. So we're going to be adding a salt solution to it. So there we go. Added some salt solution. So what the salt does, it causes proteins and other debris to clump together. So remember, we want to separate the DNA from the protein and from all of the other debris that's in there. So then it's going to go into the centrifuge here. And we have to put, so in order to balance it, we have to put a tube with water on the other side. And then let's close it and turn it on. So what happens inside the centrifuge is that they spin around in a circle really, really fast. And you can see here that all the protein in that, they like collapse to the bottom, while the DNA stays in the liquid that's above here. So we're going to take the liquid that has the DNA in it, and we're going to remove it from this one little tubey thing, and we're going to put it into another tube. So basically we're isolating the liquid from the solid that was in that tube. Now what we're going to do is we're going to try to remove the DNA from the rest of the liquid that's in there. So we're going to add some alcohol. So you can see how the DNA strands, they sort of like come together like that. So because DNA is not soluble, so it doesn't dissolve in alcohol, then it's going to come out of the solution. Alright, so we're going to throw it back into the centrifuge here. And just like before, how, oh, give me one sec here. Gotta close the lid first. So like before, how we did in the last step, when all the proteins sank to the bottom, now you can see we're gonna have something else sink to the bottom here. So we can take our micropipetter here, and we can take the liquid out. I'm just going to read this out here. Once removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it, or you can move on to your next experiment. Okay. But yeah, so we take out all the liquid, and then the DNA is left in the bottom of the tube there. I think that Monique is going to be doing a little bit of a lab too. I'm not sure when she's going to do that, and I'm also not sure if that is for sure going to happen. But make sure you watch out for that. And yeah, good luck with this lab. If you have any questions, just let one of us know. Other than that, hope you guys have a good day. See you guys.